This week in the Extension Plant Pathology Lab at Montana State University, the lentils we planted are starting to emerge. The plants are growing fast, full of hope and promise. Unfortunately for them, along with the seed, we planted millet infested with Fusarium root rot pathogens. If we are successful, seeds that are not protected from Fusarium will soon not emerge, and plants will develop symptoms of root rot, including a brownish red to black discoloration of the root beginning at the seed attachment, a reduced number of secondary roots, and eventual wilting, discoloration, or plant death. We have 4 locations in Montana and 3 in North Dakota where researchers are testing whether seed treatment fungicides help prevent Fusarium root rot. Here, graduate student Sid Atencio and undergraduate Jenna Anderson are finishing the plot setup. In this experiment, we have 8 synthetic fungicide treatments, 1 essential oil, and an untreated control. The plots are arranged in 4 blocks with treatments randomized within each block. After a statistical analysis, we can tell whether seed treatments prevent root rot and whether some are better than others. Today, Sid and Jenna did stand counts. This is the number of plants that emerge in a unit area of the plot. If our infested plots resulted in disease, we would expect the control plots to have reduced emergence compared to the fungicide-treated seed. Eventually, this research will be used when we make recommendations to growers who are wondering if they should use a seed treatment, which one they should use, and if seed treatments increase yield. This experiment is just one part of a USDA Specialty Crop Research Initiative grant to help growers manage Fusarium root rot in lentil.